grateful for having this opportunity to come back virtually and share my work. It's always a great pleasure to see old friends and meet with new people. I'll try to finish my presentation in 30 minutes, and I hope to leave some time to chat with people in the end so that we can make our conversations more interactive. So I prepared one slide to summarize my research and career path. I was in Michigan from 2000 to 2005, and I worked with Mike and Gonzalo for my PhD dissertation. I started my faculty position at Penn in 2006, and I have been there since then. I'm still pretty happy here. In this slide, you can see that over the years, I have gradually transitioned my research from traditional statistical genetics to statistical genomics, and more recently to the applications of machine learning. What I'm doing right now for my research is completely different from what I did during my PhD study. But what I wanted to say is that I benefited substantially from my training at Michigan, which gave me the skills to explore different research directions. Without the training I received from Michigan, especially the advice I received from Mike and Gonzalo, who taught me how to do research, I wouldn't have gone this far. So in my presentation today, I'm going to share with you my recent work on spatial transcriptomics. In fact, most of the work is still ongoing. I think people in this audience may know that genomics is a very, very fast developing field. The type of research that we do is mainly driven by the development of technologies. In the previous slide, you may have noticed that I change my research direction about every 5 years. And that change is in fact due to the change of the technologies in the field. In the last few years, we have seen that the method of the year selected by nature methods has shifted from single-cell sequencing to single-cell multi-omics, and more recently to spatially resolved transcriptomics. I started working on spatial transcriptomics since the beginning of last year, and I found this area to be extremely exciting. I anticipate that I will continue working in this area for another 3 or 4 years. So several technologies have been developed for spatial transcriptomics. My work is mainly focused on Visium platform by 10x Genomics because this technology is commercialized and is readily accessible by most research labs in the world. As a method developer, when I decide what to work on, I tend to pick on technologies that are already commercialized because this means that my work is going to have a larger impact. This slide here shows how the Visium data look like. You can see that within each Visium slide, there are 4 capture areas, and within each capture area, there is about 5,000 barcoded spots. Within each spot, we can simultaneously measure the gene expression across all of the genes in the genome by traditional RNA sequencing. And what is exciting about the Visium data is that in addition to gene expression measurement, the Visium data also have very high resolution histology images that are generated from the same tissue sections. So the ideal method for spatial transcriptomics data analysis should incorporate both gene expression and histology image information in the data. In my talk today, I'll briefly touch upon the following questions. I will discuss how to perform spatial clustering, how to perform cell type deconvolution, how to generate the super resolution gene expression data, how to integrate the gene expression and histology to do tissue annotation at multiple different levels, and how to recover spatial location information for single cells. Well, first, I'll present SpaGCN, which is a spatial clustering algorithm that we recently developed. Clustering analysis is a very important task in spatial transcriptomics data analysis. Although many methods have been developed for clustering analysis of single cells, these methods do not consider the spatial dependency of gene expression. For data generated from a spatial transcriptomics experiment, we know that for the spots that are physically close to each other and also share similar histological features, they are also expected to have similar gene expression profiles, and they are more likely to belong to the same spatial domain. If you were to analyze this type of data using a method that does not account for spatial dependency of the gene expression, you may misassign these spots into different clusters or different domains in the tissue. So to account for spatial dependency, we chose to use a graph convolutional network-based approach because this approach is very flexible in analyzing data that have irregular shapes. A critical step when building the graph is to define the adjacency matrix, which describes the degree of similarity between every two spots in the data. To consider both spatial location and histology image information, we first define a distance metric between every two spots. And this distance metric is determined by the spatial coordinates represented by X and Y, and the histology pixel intensity represented by Z. By considering the histology as the third dimension in the Euclidean space, we can calculate the Euclidean distance between the two spots represented by U and B here. And once this distance metric is defined, we can further calculate the edge weight between the two spots, which is defined as an exponential decay function of the Euclidean distance. And this graph can be represented by the adjacency matrix that contains the edge weights between every two spots in the data. After this graph is built, we then perform graph convolution to aggregate information in gene expression across different spots. And this graph convolutional layer is then further connected to an iterative clustering layer in order to get a cluster assignment for each spot. In the end, we can get a cluster assignment for all of the spots in the data, and we call each cluster as a spatial domain. The domains that we infer from the previous clustering analysis allow us to perform domain-guided differential expression analysis in order to identify what genes are spatially variable. Spatial variable genes refer to those genes whose expression pattern shows variation in the space in the tissue. So for example, for this orange domain here, which corresponds to the cerebellum of the brain, we can identify a gene that is shown here that uniquely marks the expression pattern of the cerebellum of the brain. And sometimes we see that when you analyze the real data, some of the spatial domains have very complicated tissue structure. It is not possible to identify single genes to represent the spatial expression pattern of those domains. For domains like those, we have an automatic procedure which allows us to infer meta-genes such that the combination of multiple genes can mark the expression pattern of a given domain. And here is one example meta-gene that we can infer from our SpaGCN algorithm. To evaluate the performance of SpaGCN, we analyzed many data sets. Here, I just show you one application to a data set that is generated from mouse posterior brain. We analyzed this data set using 4 clustering algorithms, including Louvain, ST-learn, Bayes space, and SpaGCN. Louvain is a commonly used clustering algorithm that people use for single cell data analysis, whereas ST-learn and Bayes space are both recently developed methods for spatial transcriptomics clustering analysis. From these clustering results, we can see that Louvain's cluster is similar to ST-learn, Bayes space, and SpaGCN, but the spatial domains detected by the other 3 methods are more spatially contiguous due to their ability to account for spatial dependency. So after seeing this, next, we wanted to ask, what is the ability of each method in detecting more refined tissue structure? And specifically, we performed subclustering analysis for spots in domain 5 detected by SpaGCN, which corresponds to the cortex of the brain. So here are the subclustering results obtained from each method. This is the annotated result obtained from SpaGCN subdomain clustering analysis. By comparing the subdomains obtained from SpaGCN and the tissue layer structure annotated by the Allen Brain Atlas, we can see that the SpaGCN results agree almost perfectly with the annotation provided by the Allen Brain Atlas. So for example, this subdomain 0 corresponds to layers 2 and 3, and this subdomain colored by purple domain 2 corresponds to layers 4 and 5. In addition, we detected layer 6, the cerebellum region in the brain colored by blue, and we also detected the hippocampus CA1 region in the brain. And more importantly, we see that the cluster results obtained from SpaGCN are better than the results obtained from the competing methods. So for example, when you look at the Louvain and the ST-learn cluster results, you can see that they mixed the cortical layers together into one spatial subdomain, which do not agree with the cortical layer structure in the brain provided by the Allen Brain Institute. Bayes space performs slightly better than Lovin and ST-Learn. However, Bayes space clustered layer 6 and the hippocampal CN1 region together as one subdomain, and this clustering result is not correct. So overall, this analysis indicates that our SpaGCN algorithm has higher sensitivity in detecting more refined tissue structure. SpaGCN is also able to detect the spatially variable genes. Here I show you a few example genes that we detected. So for example, for domain 1, which is colored in yellow, we detect this gene PVALB, whose expression pattern marked perfectly for this domain. And for domain 5, we detect the gene NRGN. You may have noticed that this gene is also expressed in domain 7. The reason is that the tissue section that we work with only represents a cross-sectional cut of the brain, but the brain itself is 3D. In the original brain, in the 3D space, domains 5 and 7 are in fact connected with each other. They belong to the same spatial domain in the brain. So it's not surprising that we are able to identify a gene like the NRGN for domain 5. For domain 8, we infer the gene TRIM62. You can see that this pattern is very clear for domain 8. And if you compare domain 1 and domain 8, you can see that they are adjacent to each other in the brain. But using our approach, we are able to clearly tease out what genes are specific for domain 1 and what genes are specific for domain 8. For domain 0, which is colored in red in this plot, you can see that this domain has a very complicated structure. For this domain, we are not able to identify a single gene to represent the spatial gene expression pattern. However, we are able to identify a metagene that is formed by 3 genes, KLK6, MBP, and ATP1B1. By combining the gene expression provided by KLK6 and MBP and subtracting the gene expression of ATP1B1, we are able to come up with a metagene such that this metagene can uniquely mark the spatial expression pattern of this complicated spatial domain 0. Although Visium is a very powerful technique, a major limitation of the current Visium platform is that it does not have single cell resolution. So in order to fully understand how different cell types are distributed in the tissue, we need to integrate with single cell RNA sequencing data and perform cell type deconvolution analysis. But in the next part of my talk, I'm going to present SpaDecon, which is a method that we recently developed for cell type deconvolution analysis. The key idea of SpaDecon is to perform gene expression smoothing so that we can utilize spatial location information as well as the histology image information. Our intuition is that for spots that are physically close to each other and also similar to each other in histology, they should also have similar gene expression. So this means that we can borrow information from similar spots and perform gene expression smoothing. And this will make the gene expression information more reliable and more spatially coherent. To smooth the gene expression, we multiply the original gene expression data with the adjacency matrix derived from SpaGCN. And after this gene expression is smoothed, we then use a transfer learning-based algorithm to infer cell type proportions. And here, we assume that there is a single cell reference data set where the cell types are already annotated. We start by building a network to extract the cell type specific gene expression signatures from the training data. And we then transfer the weights learned from this training data set onto the target network that is based on the smooth gene expression in spatial transcriptomics data set. And this target network is then further fine-tuned so that the cell type specific gene expression signatures in the spatial transcriptomics data can be captured. After this fine-tuning step is finished, the target network then returns a cell type assignment for each spot. We interpret the cell type assignment probability as the corresponding cell type proportion. So to evaluate the performance of SpaDecon, we analyzed a spatial transcriptomics data set generated from mouse brain. We used a single cell RNA sequencing data set generated by the Allen Brain Atlas as the reference. We compared with a few other competing methods for cell type deconvolution analysis of spatial transcriptomics data. And these methods include RCTD, which is recently published in Nature Biotech, stereoscope, and spotlight. These plots here showed the deconvolution results. For each spot, we displayed the estimated cell type proportions as a pie chart. By comparing these plots, you can see that using SpaGCN, we can clearly infer the cortical layer structure in the brain. You can see the gradual change of layer 2 to 4, 5, and 6. And furthermore, we inferred where the oligodendrocytes are located in the mouse brain. This region corresponds to the white matter region of the brain. But if you look at the results inferred from the competing methods, you see that they mixed the cortical layers structure, and some of the spots they cannot infer very well. So next, we analyzed a complicated cancer data set. This one is from a melanoma patient where the authors generated spatial transcriptomics data for the tissue sections obtained from a melanoma patient. And this plot here shows the pathologist annotated tissue, where the black region represents the melanoma region of the tissue, the yellow region represents lymphoid tissue, and the red region represents stroma. But looking at the results obtained from SpaGCN, we see that we are able to infer T cells who are located near the tumor boundary. And furthermore, we detected the presence of T cells and the B cells at the bottom of the tissue and also at the upper right part of the tissue. The locations of the T cells and the B cells that we inferred agreed very well with the pathologist and the annotated lymphoid tissue. By contrast, if you look at the results inferred from the RCTD, you can see that it inferred a very little presence of T cells in the tumor boundary, and also at the bottom of the tissue, very little presence of the T cells. And at the upper right corner, it even inferred some malignant cells in a few spots. But based on the pathologist annotation, this region of the tissue does not contain any tumor cells. Compared to RCTD, the performance of a spotlight seems to be even worse. The performance of a stereoscope in general is pretty similar to RCTD. But one thing I want to comment on is that stereoscope is extremely slow program. It's not computationally feasible for large-scale data sets. The cell type deconvolution analysis can tell us how different cell types are distributed in the tissue, but still doesn't tell us the cell type level gene expression. So in the next part of my talk, I will briefly describe a super resolution gene expression algorithm that we recently developed, which can substantially increase the gene expression resolution. So I have mentioned earlier that Visium data do not have single cell resolution. In addition to the lack of a single cell resolution, there is a lot of tissue gaps in the Visium data. And these tissue gaps are not covered by spots. But on the other hand, we have the companion very high resolution histology image that is obtained from the same tissue section. So our goal is to utilize the high resolution histology image data to enhance gene expression resolution and impute the gene expression in the tissue gaps. To do so, we use a contour detection algorithm to first detect the tissue area. And after that, we generate a very fine grid of the tissue and impute the gene expression for each superpixel within the tissue area. To impute the gene expression for each superpixel, we identify its nearest neighboring spots using the edge weight defined in SpaGCN, and we then use their weighted average gene expression to represent the unobserved gene expression at the superpixel. So using this approach, we analyzed the mouse posterior brain data set, and here are a few example genes that I picked. The first row shows the original observed gene expression, and the second row shows the enhanced resolution gene expression. Comparing these gene expression pictures, you can see that our algorithm can substantially increase the resolution of gene expression, and there's no tissue gaps in the data anymore. Using our super resolution gene expression algorithm, we can convert the gene expression into an image. And this opens the door for many interesting imaging-based data analysis. And next, I'm going to show you how to integrate the gene expression and histology images together for tissue annotation. For this application, I'm going to use cancer data as an example. To annotate the tissue, we first generate super resolution gene expression images for a set of marker genes. These marker genes are provided by users. So for example, if you wanted to study tumor, you may provide a list of marker genes that are specific for tumor. And if you wanted to study a particular cell type, for example, immune cells, you may submit a list of cell type marker genes for a particular immune cell type. And because the different cell types may have different numbers of marker genes, in order to make our method robust to the number of marker genes, we chose to use a meta-gene to summarize the gene expression pattern for each cell type. So this way, we only have one single gene expression image for the corresponding cell type. For the histology, there are 3 color channels, RGB. In order to make the histology image comparable to the gene expression images obtained from a meta-gene, we convert the original histology color image into a grayscale histology image. And after this conversion, we have one gene image for each cell type and one image for the histology. And we can treat the gene expression data as an additional color channel in the histology image to perform joint segmentation using a convolutional network-based approach. So for example, when combining the gene expression provided by tumor-specific marker genes, we can detect where the tumor is located in the tissue. And when combining the gene expression provided by cell type-specific marker genes, we can detect where a specific cell type is located in the tissue. So for example, when your goal is to infer where CD4 T cells are located, you can submit a list of marker genes for CD4 T cells, and we can directly tell you where the CD4 T cells are distributed in the tissue. After we detect the tumor region, we can further automatically detect the tumor edge and the tumor core. And by performing differential expression analysis between the edge and the core, we can detect what genes are enriched in the edge and what genes are enriched in the core. This type of analysis will allow us to have a better understanding of the intra-tumor heterogeneity. Next, I'm going to show you that we can also computationally detect the tertiary lymphoid structures in cancer. The TLS are abnormal lymphoid tissue that resembles secondary lymphoid organs in our body, and they are believed to be the site of immune response activation against the tumor. Numerous studies have shown that. In the TLS, we typically see an aggregation of B cells and CD4 T cells, and a little bit of dendritic cells nearby. But when using the marker genes for B cells, CD4 T cells, and the dendritic cells as input, we can detect where these different cell types are distributed in the tissue. And by further co-localization analysis of these different cell types, we can infer where the TLS is located in the tissue. So for example, here we detected a TLS. And to confirm if this is a true TLS, we zoomed in the tissue region in the histology, and our pathology collaborator confirmed that this is a real TLS in the tissue. So far, I've talked about spatial transcriptomics-centric analysis. In the final part of my talk, I'm going to discuss how spatial transcriptomics data can help recover the spatial locations of single cells. So we know that single-cell RNA sequencing is a very powerful technique for characterizing cell types and the cell states. But a big limitation of single-cell RNA sequencing is the lack of location information. Because once the cells are dissociated from the tissue, we lose the location information of each individual cell in the original tissue. But on the other hand, people are starting to generate a spatial transcriptomics data where we simultaneously measure gene expression and the location information. So our goal is to utilize information learned from the spatial transcriptomics data to help us recover the location information for single-cell RNA-seq. So when you recover the location, there are two possibilities. You can either recover the exact location of a cell in the tissue, or you can lose your resolution, just recover the corresponding tissue layer of the tissue, for example, cortical layers, if you study brain. To exactly recover the location of the cell in terms of the XY coordinates in the tissue, that is an extremely challenging task. Because in order to precisely recover the location at that level, you have to have reliable spatially variable genes that can tag the spatial variation of the genes in the tissue. And so far, people don't really know how many such genes exist. the presence of these genes, the information could be very, very limited. A more feasible task is to recover the layer structure of the cells in the tissue. So this slide here shows an overview of our location recovery model. The key idea is to use the spatial transcriptomics data as the training data to learn the relationship between gene expression and spatial location. We use the model learned from the training data to recover the exact location or the tissue layer for the cells in the single-cell RNA-seq data set. And because the training data set might be small, because it's still pretty expensive to generate data from the Visium platform. In order to get enough information in the training data, we also have an optional data augmentation step where we use a generative model, a variation autoencoder approach to generate the spatial gene expression data. But this way, we can enlarge the sample size in the training data set. To learn the relationship between gene expression and spatial location, we use a supervised feed-forward neural network. For the exact location recovery, we use a mean squared error loss. And for tissue layer recovery, we use a loss function that is inspired by rank consistent ordinal regression. We applied our tissue layer recovery model to a single-cell RNA-seq data set generated by Hodge et al. This was published in their 2019 Nature paper. In their study, they manually dissected the 6 cortical layers from postmortem human brains and generated single-cell data from each layer. Our goal is to computationally recover the layer information for the single cells in this data set. Because the true layer information of each cell is already known, this data set allows us to evaluate our layer prediction accuracy. We train our prediction model using the spatial transcriptomics data set generated by Maynard et al., where the authors manually annotated the cortical layers for each of the 12 tissue sections that they generated from this study. Using the information learned from this spatial transcriptomics training data set, they predicted the layer information for cells in the Hodge et al. Study. And these bar plots here show the results obtained from our method and a competing method that is called a tangram. This red bar here indicates the percentage of the cells that are correctly predicted, and the pink bar indicates the percentage of the cells that are predicted to a tissue layer that is immediately adjacent to the true tissue layer. Using our approach, we can achieve an overall accuracy of 41%, and we noticed that for many of the cells, about 35%, we cannot correctly predict their true tissue layer. However, we are able to predict these cells to the immediately adjacent tissue layer. And for the competing method, the tangram, we see that its accuracy is very low. It's only 16%. It's barely better than a random assignment of a cell to the tissue layer. I think part of the reason for the poor performance of tangram is that tangram aims to recover the exact location of the cells in the tissue. And as I have mentioned in the beginning, this is an extremely challenging task. But overall, our results indicate that it is possible to use the spatial transcriptomics data to help recover spatial location information of single cells, at least at the tissue layer level. In addition to the problems that I presented in the previous slides, I also work on some other related areas that are essential for our understanding of human disease. And these include the digital pathology, spatial metabolomics, and ultimately, I think we should think about how to integrate different types of omics data and the digital pathology data together to have a more comprehensive understanding of human disease. And such type of research would require expertise in genomics, pathology, imaging, and machine learning. I found this to be a very, very exciting area of research to do data integration and to learn information from digital pathology and other aspects of the tissue. And I think the methods that we develop for these all sorts of data integration can be eventually applied to study many different human diseases. Finally, I would like to thank members of my lab, especially Jian, Kyle, and Qi Huang. My work was based on hard work done by them. I'm very lucky to have opportunities working with these very talented young researchers. I also want to thank my wonderful collaborators. Eddie and Dave are neuropathologists at Penn. I received a lot of input from them on pathology. Taki is my Penn colleague, biostatistics professor. I received the input from him on imaging data analysis, which is essential when we started working on the histology image data. And Linhua and Human are my cancer biology. From them on cancer biology. Thank you all for listening. I'm very happy to take any questions that you may have.
Thank you so much. Thank you. Yeah, it shows that the internet is not stable. I hope you guys can hear me okay. But thank you so much, Mingyao, for the great talk. And any questions related to the research? Mike, you have some comment about the research?
No, I think it's just great and quite a departure from when Mingyao was a student in the department long ago.
Yeah, I graduated a long time ago. I graduated in 2005. I've been working at Penn for 16 years. Wow. It's a long time.
It's been a while.
Mingyao, I have a question. Peter Song. Nice to see you. Very nice talk. And in general, do you have any comments how large the training sample data is required to produce reliable learning results? I think you have developed and applied different methods, and like transform learning, you need to learn a kind of weighting of network structure and transform the weight to another sort of different data set, and to sort of propagate the results to generate new knowledge. But the question here is, I have seen a lot of this sort of transform or results from your 20 data set that I wonder about the reliability and what's the error bound that we expect in this kind of analysis.
I would say the smallest data set we tried has a few hundred cells. So the sample size is a few hundred. I think that is okay to start with. But in general, I think having larger training data set is essential to get a good performance for this type of machine learning based methods. Sample size is never an issue for the problems I work on, honestly. So I don't consider this as a big problem. Because when we work with a single cell RNA sequencing data, typically the number of cells we work with can be more than 10,000. It can be easily larger than that. And for the spatial transcriptomics data that I work with, I mentioned that the number of spots in the Visium platform is about 5,000. But of course, the total number of spots that cover the tissue area is within like 2,000 to 4,000, depending on the type of tissue that you study. And for the melanoma data set that we analyzed, we only have 300 spots or even less than that.
So Mingyao, you mentioned that you have been working on statistical genetics and then move on to the genomics research. I know many of our students have been working on these fields. And do you have any suggestions or guidelines toward to these fields for them?
I think my suggestion is to read broadly. When I was a student, I remember I read papers every day. Yeah, and I like to discuss the papers I read with Mike and Gonzalo to get their feedback and the suggestions. In the beginning, I like to read the papers that are very mathematical, but I don't read those papers anymore nowadays. Now I read papers in Nature Science, Cell, Nature Biotech, Nature Methods, genome research, genome biology, these type of journals. The reason I read these journals is because I want to see what is going on in the field. What are the problems that people care about? So in the beginning of my presentation, I showed that Nature Methods select the method of the year each year. In 2013, they select single cell sequencing as the method of the year. I noticed that and immediately I thought I should start working on single cell sequencing. So you'll see that I started working on that in 2015. I attended my first meeting on single cells, I think in 2014. And after that meeting confirmed that I should start working in this area. So I have been working on single cells for about 5 years. And honestly, I got tired of doing single cells because it's not challenging to me anymore. I'm the type of person who enjoy learning new things. So around 2019, I realized, single source is not enough. We should do, we should do more beyond single source. And that time I started to think about spatial stuff, because the obvious next step is to consider the spatial location information. So I started working on spatial stuff around the beginning of 2020. That started from a project with Jian. At that time, he was looking for a dissertation topic for his second dissertation paper. We read many papers, and we finally decided to work on spatial transcriptomics. And then Kyle started doing a rotation with me, and I asked him to do a cell type deconvolution analysis with me. So these two students helped me to get started in this very exciting area. And then after I started working in this area, the Nature Method selected spatially resolved transcriptomics as method of the year. And I was like, oh, I picked the right direction, even before they announced spatial transcriptomics as method of the year. So I think my general advice to the PhD students in the program is that don't just restrict yourself to what you have learned. I think the PhD training only trains you how to do research. I benefited substantially from my conversation and the training advice I received from Mike and Gonzalo. Mike gave me a lot of freedom to explore different research areas. I read papers every day. I think that built a very solid foundation for me to do my research. I noticed that a lot of PhD students, they only read papers that are directly related to their current project. I do not think that is a good practice.
Yeah, so read broadly and think adaptively. Also try to pick up new challenges. Those are very good advices to us, I think. So with that, I will stop the recording and open the floor for the free chatting, and any questions, please throw out.
Yeah, I thought